Okay, so now that we have a good idea of the components of the cell, the organelles, the things that make up the cell and the functions, individual functions of the organelles and things, we're now going to switch and talk more specifically about the processes going on within the cell. And we're going to start by talking about the use of energy, which occurs in the mitochondria and cytoplasm of cells. But we need to back up and first talk about energy, how it's used, and some of the fundamentals. And this is the study of this energy changes is called thermodynamics. So this is a branch of chemistry which looks at energy, its changes of state, and thermodynamics affects how living things can use energy. And so it's very important to understand some basics to understand how energy is consumed in a cell. So there are two forms of energy: kinetic energy, which is energy in motion. So that is energy being used. So for example, I am using energy to project my voice, to make my vocal cords vibrate, to produce sound. Okay, potential energy then is stored energy. So the energy that I'm using comes from energy that I have stored in the form of bonds within molecules of glucose or fat or other energy molecules. So energy can take many different forms: mechanical energy, heat, sound, electric current, light, or radioactivity. It's often measured in heat or a change in temperature. So for example, the calorie, which is how we measure the energy in food, is the amount of energy required to increase the temperature of 1 gram of water 1 degree Celsius. So where does all the energy come from that sustains life and living things? We talked about this before. Ultimately, it comes from the sun and its constant radiation towards the earth. As it does so, it's captured in living organisms through the process of photosynthesis. So plants, cyanobacteria, algae are photosynthetic and can capture light and turn it into energy in the form of bonds between molecules. It's stored in living organisms then again through sugars such as glucose, or you may string some sugars together to form glycogen or starch. Or if more long-term energy storage needs, it's in fat and lipids. And we use the energy then after it is stored by breaking down these bonds, and as we break them down, it is gives off energy. And the usable form of energy in living things is through the breaking off of a phosphate from adenosine triphosphate, or ATP. So that's the usable form in energetic processes within a cell. So we're talking about energy being gained and lost through bonds. This occurs through oxidation and reduction reactions. These are always paired, meaning for every oxidation, which is where you lose an electron or lose a hydrogen, you have another molecule being reduced, which is gaining an electron or gaining a hydrogen. When a molecule is reduced, it now has more energy because it gained an electron, it gained a bond, gained a hydrogen, and thus is better, has one more bond than it used to. So these reactions within a cell all fall subject to certain laws which are universal. The first law of thermodynamics is that energy can not be created or destroyed. It can only be changed from one form to another. If you remember before, we talked about the different forms, including light, sound, heat, electricity, and so forth. So the first law of thermodynamics, you can as this bear is doing here, eat food and gain energy, and that energy may change forms from mechanical to chemical and so on and so forth and back and forth. But the amount of energy in the universe is constant, is finite, is all that's there is there, and there's not going to be any more or less. When energy is converted from one form to another, some of it is lost as heat. So there is no 100% efficient transfer from one form of energy to another. So this relates to the second law, which is that in the transfer of energy, that heat that is lost adds to the random order of matter. And this is continually increasing in the universe, meaning everything is becoming much less ordered and much more random. The term to describe this is called entropy. So entropy is always increasing in the universe, especially in the transfer of energy from one form to another. So for example, here this bear is turning chemical energy that he got from that fish he ate into mechanical energy as it is actually running through the contraction of muscles. And as it does so, it's losing some forms of energy in the form of heat. It will start to overheat if it does this for too long. That heat is adding to the disorder of the universe. So we have ways to measure this, and we use symbols to describe different things. So the capital G is a representation of free energy, or the energy available for work. So if you remember, in every conversion you lose some to heat, well, then free energy is the amount that's actually available. So it equals H minus Ts. H is enthalpy, or the energy within the bonds of a molecule. So this is the potential energy, the full potential energy available. But when we use it, some of it is lost as entropy, and that depends on at what temperature it is being used. So temperature times entropy subtracted from enthalpy equals our free energy. In a reaction then, we have a change in free energy. So free energy goes from one form to another. If our change in free energy is negative, then that reaction is exergonic. Okay, so that's what we have here. In an exergonic reaction, the reaction releases energy as part of it. And it is spontaneous, meaning you don't have to input any energy, it just happens automatically. On the other end, if you have the products gaining energy, or a reaction requires energy to complete, this is called an endergonic reaction, and that is when your change in free energy is positive. This is not spontaneous. It requires, again, an input of energy in order to do that. So, in an exergonic... so this is similar to just an analogy. Watching lecture requires energy, requires you to actually study and listen. Watching Netflix, however, is like an exergonic reaction in which you just sit there and is spontaneous, doesn't take any amount of energy to do it. All right, in an exergonic reaction, you have a certain amount of energy that is required for the energy to go to completion, and that is called the activation energy. And the activation energy then affects the rate of the total reaction. And you can increase then the rate of the reaction either by increasing the energy so that you can overcome that activation energy faster, or you can decrease the activation energy. Well, how might you decrease the activation energy? Well, that is done through what is called a catalyst. And a catalyst affects the chemical bonds in a reaction so that the activation energy is lower. So it facilitates the reaction. However, they do not change the products or the reactants, and they also don't change themselves. So it's really exactly that. A facilitator takes in the reactants, allows them to be reconfigured in a way which is favorable, and then causes the reaction to occur. Again, like we said before, if this black line is the normal spontaneous exergonic reaction, then you are it is occurring at a specific rate. But if you decrease the activation energy with a catalyst, you can, which is an enzyme is a catalyst, you can increase the reaction rate. We mentioned this before. Adenosine triphosphate then is the molecule used to store and use energy in the short term. So when you want to actually use energy, you use ATP. It is made of 3 components: the ribose, which is the 5-carbon sugar, right? So this is the same 3 components which are for nucleotides. So this is a nucleotide. So the 5-carbon sugar, it has adenine, which is the base, and it has 3 phosphate molecules. So this is the phosphate groups. Now the number of phosphate molecules is equivalent to the amount of potential energy that is on there, because these bonds are unstable, so you can add a phosphate relatively easily, but you can also remove a phosphate relatively easily. So if you take off one of these phosphates, then you have adenosine diphosphate. If you take off another one, you have adenosine monophosphate. As you take them off, you release energy, and that energy can be used for metabolic processes. So ATP, again, is used through the process of hydrolysis, so adding a water and causing the bond to break. It doesn't store very well long-term because of these unstable bonds. So if you want more long-term energy, you're going to use glucose, glycogen, starch, lipids. All right, so this is a 3D model of sorts of an enzyme, and you can see its different parts. You have the substrate. This is what's going to react, go through a reaction. The active site, and these enzymes are usually made of protein. The substrate will then bind to the active site, and then it will reconfigure that substrate in a way which makes the reaction occur faster by lowering the activation energy. After the reaction, the substrate will move on, and the enzyme will be able to be reused in the reaction. Okay, so again, here's the enzyme. When it binds to the substrate, it forms the enzyme substrate complex, applying stress to the bonds, changing the shape, and lowering the activation energy. I know this is redundant. Okay, so here is the multiple steps in which you have it. You have the substrates entering the active site, forming the substrate, enzyme substrate complex. The substrates are then converted into the products. The products are released, and now the enzyme is available for reuse. All right, now enzymes work well under a range of conditions, but it has an optimum range of conditions where it will work best. Okay, and these can change based on temperature, concentration of reactants, products, and enzymes, the pH, and also salinity. There are also other things which may affect the reaction rate of the enzymes as well, including the presence of inhibitors or facilitators, but we're just going to talk about inhibitors at this point. These bind to the enzyme and decrease its ability to catalyze a reaction. We talked about these in lab. There are 2 different ways in which it can bind to decrease the enzyme's ability to affect a reaction. The first is a competitive inhibitor. This is something that binds in the same site as the substrate. And so when it is bound there, the substrate cannot bind and it cannot react. A non-competitive inhibitor then will attach at a different spot, and it will change the shape so that the substrates can't bind. Allosteric enzymes are enzymes that have an inactive form. Now, the inactive form may be because of an inhibitor or an activator. In an allosteric inhibitor, when it is bound at the inhibitor site, the shape of the enzyme changes so that it cannot be used. If it's an activator, in order for it to work, the allosteric activator has to be bound, and then it can attach to the substrate. Now, why are all these things important? Well, metabolism is the total chemical reactions that take place, and many times it'll have a cascade of reactions that have to occur. Some of these reactions may be anabolic, which means that energy is used to build molecules. These are generally endergonic reactions. They require energy because you're building something, making something more complex. Or you may have catabolic reactions where energy is being used as you break down molecules. Biochemical pathways then, such as the one down here, involve many different reactions, and each product being the substrate in the next step, until you get a final product. Now, this final product also may be important for the regulation of its own product, and that may be done through a process called feedback inhibition. We don't want these biochemical pathways to just be constantly going without any regulation, and so one of the ways is through the product. You can also bind back to the enzyme, causing inhibition through allosteric inhibitor. And so now, if you have too much of isoleucine, such as in this example, it will bind to the enzyme and prevent more production of isoleucine. And in that way, you can regulate how much isoleucine is being produced, and overproduction is avoided. Okay, so now that we've done the background of energy and enzymes, now we're going to talk about what specifically in the mitochondria is occurring to form energy. And this occurs through the process of respiration. Respiration is converting organic molecules, breaking them down, and using oxygen to make ATP, usable energy. Autotrophs are organisms that are able to build molecules, make anabolic reactions to form bonds that can later be used to make ATP. And these are ones that photosynthesize. Heterotrophs rely on other organisms, so eat other organisms and then break down those molecules to allow the process of respiration to occur and energy to be used. So, cellular respiration is a series of reactions starting with an organic molecules. We're going to talk about glucose, and we'll talk about other substrates which can also be used for energy. And the whole purpose of this is to make ATP, and ATP can then be used. Now, these are vital for creating what's called a proton gradient, and we'll see this later. We have redox reactions where things are being reduced and things are being oxidized in order to form molecules which can make a proton gradient. As you remember before, an oxidation reaction is where you lose an electron or lose a hydrogen, and in a reduction reaction you gain an electron or you gain a proton. Dehydrogenation then is a specific type where lost electrons are accompanied by protons, which thus makes a hydrogen atom. All right, so an example of a redox reaction which occurs multiple times through cellular respiration is the reduction of NAD+. Okay, so NAD+ in this reaction is gaining a hydrogen, and so it was reduced when going from left to right. This other molecule here, when going from left to right, it lost its hydrogens, and so it's becoming oxidized. Okay, so this energy-rich molecule is being oxidized, going from left to right. Going from right to left, it's being reduced. NAD plus, on the other hand, is being reduced going this way and oxidized going that way. So the sequence of redox reactions in cellular metabolism oxidizes organic compounds, so these are the ones that are donating energy, reduces electron acceptors such as NAD plus and FAD plus to NADH and FADH2. So now they have extra hydrogens where they didn't have them before. So now there's potential energy here in these bonds. You then take these and oxidize them to create protons on one side of this membrane. In that process, you reduce oxygen to make it into water. That occurs right there. And then once you have all these protons over here, you can then pass them through what's called ATP synthase to make ADP and phosphates into ATP. That's the quick story. We're going to go over this in a lot more detail. In aerobic respiration, oxygen is the final electron acceptor. In anaerobic respiration, which occurs in some bacteria in hydrothermal vents and extreme environments such as in geysers, it's something else besides oxygen, right? In fermentation, the electron acceptor is an organic molecule. This is done by most bacteria. Can also be done by eukaryotes when oxygen is not available. We'll talk more about that as well. So there is a lot of free energy in glucose, 686 kilocalories per mole. This can be even higher if the glucose is already in the cell. So this large amount of energy, if it was released all at once, would be quite explosive and not very useful. But if we can do it in lots of different steps, which is what this figure is showing, you can take each of those steps and make ATP and then harness those ATPs to make available for energy. This is the equation for cellular respiration. You start with glucose, you add oxygen, and you end up with carbon dioxide, which is waste, and water. What you don't show in here is also you have a lot of ATPs which come out of it. So ATP has much lower free energy at 7.3 kilocalories per mole. And there are two mechanisms for making ATP. One is called substrate level phosphorylation, and that's basically using organic molecules such as glucose and in the citric acid cycle to make some ATP. And this is done, and it makes some ATP, but not very much. Most of it, however, is done through a process called oxidative phosphorylation, which is the last little bit. So this is where we make the proton gradient. We pack a bunch of protons on one side of a membrane, and then as they diffuse through, we can use that energy of them diffusing through to make ATP. Again, we'll go over this in detail, starting with the oxidation of glucose. Okay, there's 4 steps. The first step is glycolysis. The second step is pyruvate oxidation. The third step is the Krebs cycle or the citric acid cycle, and I'll use those interchangeably. And then the last step is electron transport chain and chemiosmosis or oxidative phosphorylation. All right, so let's start with glycolysis. This converts one glucose into 2 pyruvate molecules. And we're just going to go through the basics. When you get to biochemistry, you're going to talk about all the different enzymes and intermediates between these. But we're just going to talk about the beginning and end products. So as you do this, so glucose is 6 carbons, pyruvate is 3 carbons. So you make 2 pyruvates with 1 glucose. As you do so, you reduce 2 NAD pluses to NADH. And so now you have some potential energy there. And you have 2 ADPs, which are made into ATP. Now, in order for glycolysis to continue, you need NAD+. Okay, so you have a couple other couple options for doing that, which we'll get back to. NADH you can just pass it through the Krebs cycle and the electron transport chain to make NAD+. Or you can use a process called fermentation, which uses organic molecules to make NAD+ by reducing pyruvate and thus oxidizing NADH. But we'll talk more about that later. The next step then is pyruvate oxidation. In the presence of oxygen, pyruvate is then oxidized. Okay, and so it actually loses a carbon and forms this 2-carbon acetyl-CoA. It forms another NAD, or reduces another NAD+, forms a carbon dioxide, which is waste. And then this acetyl-CoA can then be used in the Krebs cycle, also known as the citric acid cycle. So in the citric acid cycle, you have that acetyl group oxidized from pyruvate. This occurs in the matrix of the mitochondria, and there are 9 steps. We aren't going to go over all the details and the enzymes and everything used, but just know these 3 major parts. The first part is acetyl-CoA is added to oxaloacetate, which is a 4-carbon sugar and a 4-carbon molecule and a 2-carbon molecule, to make this 6-carbon citrate. And so this is also known as citric acid, and that's why it's called the citric acid cycle. Okay, so after you have citrate, then you can rearrange and decarboxylate, which means take off carbons in the form of carbon dioxide. So you can see that happens here and here. And then you're getting you're reducing NAD pluses into NADH in a couple spots there. You're forming an ATP here, and then you also get a reduction of FAD plus there. And the final step, the last, is you're going to regenerate oxaloacetate so that you can form citrate in the next step. So those are the 3 main steps, and we're not going to go through all these individual 9 steps. So for each acetyl-CoA, and remember we had 2 pyruvates from 1 glucose, and so we're going to have 2 acetyl-CoAs for 1 glucose. You're going to have 2 molecules of carbon dioxide, that's waste. You're going to reduce 3 NAD pluses to NADH, 1 FAD to FADH2, and have 1 ATP, and then regenerate oxaloacetate. So if you have 2 of these, and you multiply everything by 2 per 1 glucose. So at this point, if you just double all those, you'll have 6 carbon dioxides of waste, 4 ATPs of energy, 10 NADHs, and 2 FADH2s. And so now these potential energy of these NADH and FADH2 molecules are going to be put to use in our next step. So what happens next is you use the electron transport chain, which is a number of integral and peripheral plasma membrane proteins that carry electrons. And these are embedded into the inner mitochondrial membrane. And then you take your NADH and your FADH2 and you oxidize them, and you donate electrons. And as you donate the electrons, you also donate protons and pass protons through the membrane. Okay, so now you have a concentration of protons on one end of the mitochondrial membrane. At the end, you have oxygen, which acts as the electron acceptor. As it accepts those, it forms water. So then the last step is, now that you have this accumulation of protons from our proton pumps, those now want to diffuse to the other side of the membrane where there aren't as many protons. So they diffuse through what's called ATP synthase. As they do so, the energy of them passing through is used to form ATP from ADP. And so that then allows you to have an accumulation of ATPs. All these NADHs and FADH2s that we made were used, and it makes about 30 or 32 ATP. When you actually do the math, what you get is for every ATP, ATPs go straight across, but for every NADH you get 3 ATPs, and for every FADH2 you get 2 ATPs. If you added all that up, it would add up to 38. But it's not as efficient as we have shown it. So there is some leaky inner membrane where some of those protons get through and don't go through the ATP synthase. And some of the proton gradient is used for purposes other than ATP synthesis. So in reality, you get 30 to 32 ATP per glucose. All right, if you don't have oxygen, anaerobic respiration, they use different molecules such as hydrogen sulfide, or nitrate, carbon dioxide, or even inorganic metals. And these are called methanogens or sulfur bacteria. Here's an example of one here living in this hydrothermal hot springs. Fermentation then is where they use organic molecules as the final electron acceptor to regenerate NAD+. And so there are two forms, ethanol and lactic acid fermentation. Ethanol occurs in yeast. It takes the pyruvate, converts them into ethanol or alcohol, and then in that process forms NAD+. So those can then be used in glycolysis again. Lactic acid is the same thing, but it has lactate instead of ethanol. This occurs in animals, in our tissues. So when we go through strenuous exercise, we will produce lactic acid because we have exhausted the use of our oxygen and then have to revert to fermentation. Finally, other forms of macromolecules can also be used for energy. Amino acids, in order to be used, you have to take off the amino group. All those carbons can be used, but the amino group cannot be used. So it has to go through a process of eternal deamination to remove the amino group. And then it can be converted into pyruvate or acetyl-CoA or some other intermediate. Fats, what they do is you break down the fatty acids. Every 2 carbon in the fatty acid chain, you can convert it into acetyl-CoA in a process called beta oxidation. You can also use the glycerol, which is a 3 carbon, and convert it into pyruvate, and those can then be used in the Krebs cycle. So the respiration of a 6 carbon fatty acid, because it's only using 2 carbons at a time, yields 20% more energy than a 6 carbon glucose. So if you want to store energy for long term, you're going to put it in a fat. All right, finally, when you look at the whole picture here, fats can then be used as intermediates of glycolysis and or converted into acetyl-CoA. Amino acids can fit in a bunch of different spots depending on the amino acid and its structure. And then carbohydrates, of course, are converted into sugars and go through the whole process that we talked about before, including glycolysis. And that is how cells make energy.